Hello everybody and welcome back to the final video for drug delivery. This is the video covering everything for exam 3 and its topics include topicals, bioequivalence, excipients, ocular, nasal, pulmonary delivery, biologics, and patient-centered medicine. So as far as topicals go, we do need to know this chart really well. And I'm going to briefly summarize this in the video because this is kind of just something we need to memorize. But I would definitely know the 5 types of bases and generally how they work, and then as well as excipients. I actually have a Quizlet over all the excipients, every single one we've talked about in this section of the exam. So from this lecture and all the future lectures, it's about 50 slides long. I know for excipients, Dr. Lee said I believe he'd only be testing us on surfactants, binders, fillers, and I believe colorants. I have it on a note in a future slide, but I also wanted to mention it here. So it's a little bit easier than actually this entire list, but we will need to know these base types as well. So I would definitely review the Quizlet, just because I'll be mentioning the types in this video, but at a certain point we just need to memorize. So just a general rule that I do want to talk about. Creams and emulsions will actually penetrate a little more deeply than things such as oleaginous bases / ointments. This is called diadermic for penetrating deeper into the epidermis versus epidermic, which is less deep. So another benefit of like creams are spread, but they can be a little more easily removed than ointment. This can be good or bad. If it's not something that needs to be delivered too long, a cream would be fine. But if it's something like an ointment that needs to remain on your skin longer, that's when we would use something more along those lines. As far as gels, gels are kind of like water-based. They could be one or two different like phases. I would say, and this means they could have like a single gelling agent or a two-phase colloidal system. So what that would mean is a gelling agent and then something suspended kind of. Gels are usually more water-based. They can be alcohol or propylene glycol are two really common kinds of bases for gels. Paste are a little less common. Uh, we, I believe we compounded paste in Skills Lab, or at least something that would be the equivalent of a paste. Generally, paste will have larger insoluble agents, and these will be solid. So you can think of like metals or really large molecules like starch. And these will be like harder to spread. They'll be a little bit drier generally, um, and they'll be pretty stiff overall. Um, but those are not like official classifications. Those are just kind of more general. I don't want to call them buzzwords because again, there's like overlap between all of these, but these are like general categories. They're not like these base types which have actual literal physical qualities to them. Um, and then finally, as far as FDA approval, there's actually only one FDA approved test to compare topicals. So this will almost certainly be a question, or at least come up in some form on a question. The only FDA approved test to compare topical, it actually only compares topical corticosteroids. And it's this vasoconstrictor assay. Essentially, if you were to apply a vasoconstrictor like lidocaine while giving a steroid, that vasoconstriction is going to make it so the steroid won't move as far, right? Because there's not blood flow carrying it away. Um, so you're essentially measuring the concentration of steroid after you've done this lidocaine between two samples. And if they're equivalent, that means they're bioequivalent. The FDA will allow other certain studies, but none of them are like always immediately approved like the vasoconstrictor is. You have tape stripping, which is just, I think I talked about in the last video. You kind of just rip tape off of a skin sample to look at what drugs are in the cells. There's some PK studies. Again, this can vary. There's microdialysis, so taking concentrations in like a fluid over time. But the only approved one is that vasoconstrictor, so I would definitely know that. Okay, so now let's talk about different types of equivalence. So there's 3 types, and the one we're shooting for more than anything is we're shooting for therapeutic equivalent. But I'll talk about the 2 subsections of that being pharmaceutical equivalence. All this really means, or the big takeaway, is there's the same active drug ingredient, right? They're the same compound. Um, and it's like the same dose, essentially, the same strength, quality, purity, etc. So this just means same drug, same dose. That's all pharmaceutical equivalence is. And bioequivalence is a little different. it doesn't necessarily care what the same drug is, but it cares that the rate of absorption or of the drug as well as the extent of absorption are the same. And when I say the same drug, I should specify that a little further. I believe you still need the same active for this, but you can have different inactives, or like a different kind of um, formulation. For therapeutic equivalence, we need like the same everything, right? We need both bioequivalence and pharmaceutical equivalence to make therapeutic equivalent. Because that's essentially the same effect, the same safety profile. Um, Patients are essentially exactly the same. So essentially, when we're creating a generic project or product of a topical, they must be pharmaceutically equivalent. And that is like it is the same drug, the same dose, the same purity, but we don't per se need bioequivalence. Um, right, because if you think about it, bioequivalence is just how fast the drug absorbs. But the same maximal effect is like assumed. So if the drug absorbs in a different way or at a different speed, but it does the same thing, who really cares, right? Like it doesn't really matter. There is a range that's like an acceptable bioequivalence. It must be 80 to 125%. So it's like it's got to be close, right? We can't have this drug that completely absorbs a different way, and then be considered a generic. But essentially, it's like this range of like so like 4/5 and 5/4 are reciprocal to each other, and these are respective to this 80 and 125. So that's where these numbers come from. It's like plus or minus 20 or 25%, depending on what your starting point is, to be the same kind of efficacy. So that's what you can get away with bioequivalence. How do you test bioequivalence? You can just do like fluid, right? You can do like microdialysis, like just testing the drug in the blood or in the lymph or whatever. Um, you could do pharmacodynamics. These are a little bit harder to do to prove bioequivalence because at a certain point you also need to test like absorption rate. But like again, it says like blood pressure here, right? A change in blood pressure would indicate at least the strength is the same. I would say that'd be more of a pharmaceutical equivalent test, but like you could probably set up an experiment where it was bioequivalence. I just don't know specifically how you do it. Um, or like endpoints, right? Like, does it cure the disease or not? That one's pretty self-explanatory. Um, so I mentioned this on the previous slide. For topical systems, the only way to like truly the FDA method, FDA approved method, is just that vasoconstrictor assay still with the corticosteroids. But again, more things, right? You could do like a biopsy, you can do tape stripping. I basically just repeated this from the last slide, microdialysis. So these are not FDA approved, but like as long as you set up a good assay and you sent it and submitted it to the FDA, they would probably approve it anyways. Okay, so next we have these tables that kind of show formula performance. I don't really think these are going to be asked, but I'll go over them briefly anyways. Um, they're actually basically the same for systemically absorbed products and topical products. It's just the kind of form of the drug, the formula. It's measured by dissolution of the drug. This is like true of both of them. The pharmacokinetic properties are both measured by plasma sampling. So PK samples, and then it says plasma here. It's probably like impossible to see, but that's what those are. And then pharmacodynamics are evaluated by therapeutic effects. Again, this chart's also a little out of order. Like, it's the same information on both of these, so I don't know why they switched up the order, but it's just the same thing, so don't worry too much about that. But just be aware, and it's kind of common sense, right? Like, the only way to measure pharmacodynamics is by actually seeing the effect of the drug, like, duh. Um, Here's something that will be on the exam though, almost certainly, are the like Q1, Q2, Q3 ranking system. So essentially these kind of show levels of equivalency, and most of the time a drug will only need to reach Q2 in order to be on the market, but some drugs will need Q3. Um, so I'll just go through it briefly. Q1 is just the same components, right? This means same active, this means same excipients, same everything, but the concentrations can be different. that's where Q2 varies. Same components in the same concentration, right? Same everything. If you use like methyl cellulose as like an excipient for whatever reason, it would need to be the same concentration as the actual drug. But as long as everything's still there, it doesn't really matter. Q3 is the hardest to do. And most of the time it's not incredibly practical in order to do this, especially because companies, when they patent something, might not directly put out how they formulate it to the public. It could be like a trade secret. Uh, but what you could do is you could have the same microstructure, right? So if this is like, let's say a tablet, maybe the way your excipients crystallize around your drug is equivalent. They do it in the same conformation. That would be a Q3. And then obviously these aren't for tablets. These are for like topicals and stuff. So I don't really know. I suppose there are ways to change the microstructure of a topical. I think Dr. Kumari maybe talked about that back in fundamentals, but that's so long ago I'd have to rewatch my own video on that. Um, yeah, but it's not really important how those microstructures are the same. Just if you see the word microstructure, just know Q3 basically. Um, so how can you determine Q2 kind of? Or at least how can you like compare them to each other? So one way are there's like flow test and diffusion test. So I'll write that down because I have that in my notes, but I don't think that's on the slide. Diffusion test. Right, because these, like, we need to know these components kind of, they have everything there, but like they just kind of need to work the same. It's a little more complicated for Q3 because we need to like prove this microstructure is the same, but it's also hard without like an electron microscope. No company is going to realistically do that. So what we can do in place of that to determine Q3, we could do like rheology, which is like cell studies. Same with like in vitro, right? More cell studies. Um, but even in vitro tests may not show bioequivalence because there's so many factors using like in vitro, right, is like a cell plate. So I'll write that down in case you didn't know that. Whereas like in vivo is like in an animal. It'd be great if our drug absorbs on a cell plate, but the skin is so much more complex than just a layer of cells that even these tests may not be perfect for Q3. So again, this gets really specific and really nuanced for no good reason. So that's why most of the time people just stick to Q2. And then if products differ in Q1 and Q2, you can potentially still get on the market, but you need to do way more tests. So it's usually not worth it. You usually want to at least reach Q2. Most of the time, you're most certainly reaching Q1 because I don't even know how it'd be a generic otherwise, to be honest. But yeah, just something to be aware of. Okay, so now let's talk about inactive ingredients or excipients. So these are basically anything that anything other than the active ingredient. So these do not really do anything except help absorption or they help keep the dosage form together. They just kind of exist. What do they need to be considered inactive? They can't themselves chemically or physically modify the actual drug. Right, so these can't be like, oh, when this goes into the body, it's gonna react with this drug to activate its prodrug form. That wouldn't be an inactive ingredient. I don't even know what that'd be, or if there's even a drug like that. But that's just an example, right? They're basically just inert and they don't do anything. Um, there's also sometimes where inactive ingredients can be considered active. Alcohol, for example, because right, that's gonna do something to your body. It's not like sugar, which just kind of gets like absorbed. Uh, Alcohol will, cause like CNS effects and things like that. So yeah, it can get complex, but just know inactive doesn't really do anything, right? Um, yeah, I mentioned this on the previous slide, but I do really want to stress it here. So what will be on the exam according to Dr. Lee's recitation? Fillers, binders, surfactants, which are not on this list, so I'll write it down, and colorants. These four, according to him, are the ones he has questions on. So again, I have that Quizlet. I'll post it in the description, just so you can glance at anything that's one of these four categories. But I don't think he's asking us about like glidants or lubricants, at least according to what he said. So I'm sorry if that's incorrect. Um, So let's just really quickly talk about these, right? I'm not going to go over the specific like excipients themselves, but I'll just talk about generally what they are. Um, or actually, I'll talk about like dosage forms first. So powder is like the simplest one, right? This is like just crushed up drug, right? You can put it in a capsule, you could like pill press it, you could do a lot with it, or you could just mix it with water, right? It is super simple. It tastes bad because like, you have this really bitter drug exposed to your tongue. Drugs are usually bitter because, I mean, kind of from like an evolutionary standpoint, we're not really meant to put random substance in our body. That's why we have the bitter taste to kind of be like, oh, don't eat that. And drugs are no exception. Um, yeah, but powder, a nice thing about it, you can like buy it in bulk in like stock bottles. Um, yeah, or it's good for like if you need like a huge dose that can't be in a reasonably sized tablet. Tablets, we kind of all know what these are, right? They exist. Like the second simplest dosage form. They could be like enteric coated to like absorb in different parts of the GI tract, but like very simple. Usually a drug, some binders, and like some fillers. It's not too crazy. Oral capsules, right, might have an like outer shell. Um, Usually gelatin's the most common, although hypromelose, I believe, is the I believe it's vegetarian and vegan friendly, so you might see that as well. Um, the capsules themselves are usually full of glycerin, sometimes different alcohols. Uh, yeah, that's just general. And if you may remember from Skills Lab, I believe hypromelose is usually used in suppositories a lot, because I believe it draws water. I believe it's used in like laxative suppositories, but that's more just a random fact. I don't think that'll be on this exam, so I wouldn't worry too much about that. As far as the excipients themselves, we have dilutants. I believe these are also known as fillers. In fact, I'm almost sure they are, just judging by the order these go in. Um, they are just there to add bulk, right? They don't do anything, but they are the primary volume of the tablet. Um, they must be compatible, like, duh. You wouldn't use something incompatible. Um, Binders, for example, these will kind of like be the glue that holds the drug together. It's just due to being cohesive. They're usually powdered to begin with. And it's just to make sure that everything remains intact. You don't want too strong of a binder, right? Like, because then the drug won't actually ever get out of the tablet, and it'll just pass through the GI tract. Um, yeah, and it's more efficient if it starts wet rather than dry, right? Think of it like a sandcastle on the beach. Like you need to start with like kind of damp or moist sand, and then it'll dry, and then it'll be fine. Disintegrants are what helps a drug break up in the body. So these are kind of like the opposite of binders when you think about it. It'll just help it dissolve in the stomach or wherever in the GI tract, and then things with higher surface area, right, when they're broken up, they'll absorb more. So that's kind of the purpose of those. Um, Lubricants aren't really related to the body per se. They're actually like more related to the compounding process. They reduce friction um, in like a tablet press. Um, and they're like on the outside and they allow it so it doesn't like split apart when you're pulling the press apart. Um, so again, doesn't really do anything to the body, but same kind of thing with anti-adherents. They just prevent sticking to the machine. Um, and glidants are also like a compounding one. They improve the flow, and this is kind of like, don't think like pill press, think like mass scale production, like right, like pharmaceutical companies producing these drugs. They probably got like all these machines, and to like have the powder move in a fashion that is efficient for them, they might just include this because maybe it helps the drug roll better. Um, I think I would need a chemical engineering degree to explain that fully, but yeah, that's just my understanding of it. Um, Colorants are literally just to add color and might make the drug look more appealing if it's like a pink pill versus a white pill. I don't really know. I'd say the biggest thing FDA wise is identification, right? We can't really have all our pills looking the same because that would be kind of dangerous. I'm sure those of you who've worked in community know about like look-alike, sound-alike drugs. And some bottles will even have like certain things emphasized or certain pills will be like just all those things. just don't want patients taking the same pill. So manufacturers may make the pill a different shape or make it a different color, and that's how we distinguish that. Okay, so now when it comes to manufacturing, equivalent drugs, what needs to be proven? And I think the biggest thing is that the manufacturer must be required that each unit in a batch has the same drug content on average. There's two ways to do this. There's the content uniformity test. This will essentially break up the substance and test what's in it using chemical analysis. Um, I think there's like some microscopy things that can be done as well. I think that's what the newer technologies is referring to, but I don't think that's super important. A more simple test are these weight variation tests. This is almost kind of what we do in Skills Lab when we make our own capsules. We need to see they fall in within like a certain range. Although I don't think it's as generous as 10% is. Like we use, it's probably more like 1% or even less. Um, but yeah, just to make sure these doses are uniform. Um, and as far as like actually testing these, or like these substances, we have a few of these devices here. I don't think we'll be asked about them, but here they are. There's the basket, the paddle, the cylinder, and the flow through. So just in case you see those words anywhere, that's what it's talking about. Um, Again, going back to kind of therapeutic equivalence, same thing, right? It's just the highest level. Um, Yeah, I mean, we pretty much already talked about that. Now let's move on to nasal drugs. So, nasal drugs are actually pretty good, overall. They have almost perfect, like, bioavailability. They can at least, but it's usually over 50%. One of the, other nice things that most other drugs don't have is you can do nose to brain delivery. Think of like naloxone, right? When we're having an opioid overdose, it's really nice to have that nasal spray so we can actually get in to the CNS and stop that opioid from killing somebody. Um, there's some drawbacks as well, though. These are more the benefits, right? They're just convenient, painless, blah, blah, blah. Solubility can be really hard to get in the nose. The nose isn't a very big space, so we need it to be very soluble and very absorbable. Usually you have to have really small doses, like in micrograms and not milligrams. You got to think about systemic toxicity. There's obviously genetic variability, and then you got to pick the right device, which is kind of true for every dosage form. I think overall nasal is pretty good, but just something to keep in mind. So now let's talk a bit about the anatomy of the nose. There's really two substantial areas when it comes to nose delivery, things that are actually going to make a real difference. There's the olfactory region. Um, this is kind of near the upper portion of the nose. It's about 10 centimeters squared, so pretty low compared to the GI tract, which like has meters and meters of surface area due to the cilia and how large the intestines are. And then there's also the turbinate, which is about 10 times larger at 100 centimeters. Again, still really small, but it is a thing. So it's like most of them are going to the turbinate. So I'd probably memorize that. What are the actual cells in the nose like? So we have really two types of cells that are relevant. We have like goblet cells. These will secrete like mucus, right? And the reason our nose produces mucus is because since our nose is pretty much exposed to the outside air, especially because we breathe so much, a lot of bacteria and things will get in through the air. And our nose is expelling mucus, just to kind of keep those from reaching the inner parts of our body, our blood, and then also the brain, as we mentioned, which would not be good. But we also have other areas for the brain as well. There's also these ciliated cells, right? These can also be goblet cells, but these cilia will kind of push the mucus outward through the nose to expel it. Um, and these can also be used for some drug absorption as well because they do increase the surface area of the nose. You might have been thinking before, how can this have 100 square centimeters when the nostril is so small? This is how. It's those cilia that would contribute that. Um, as far as what we need to make a good nasal drug, we need pretty much the same things as another drug. Log P and solubility are always going to be really important. The nose is unique in the fact that it has a mucus barrier, which is aqueous. So traditionally, most of our drugs have needed to be like solely lipophilic and uncharged to get into where we need them to. The nose is a little bit different, and I'll talk about this more in a second. but it also makes pH a predominant factor. I don't know why that's not on this list. It's not as big as solubility, but there are some things with pH. Um, Another thing is the drug in a solution. Is it something else? Um, Mucus can prevent drug from absorbing in certain cases, so that's something to consider. That's that mucosal barrier. So yeah, let's just go a little more deep into these properties, because we probably will get a question on this. So pH is really interesting, right, with how drugs absorb in the nose, because it's almost like we need the drug to be lipophilic, right? That makes sense with this benzoic acid. If the if it's in its acid form, right, it's uncharged. It's that HA form, and its conjugate base would be A minus. And remember that charged substances wouldn't want to cross in to like a transcellular pathway like this. But it's interesting because with this mucus layer, if you just think about things that are soluble in water, like electrolytes, things that have charge, it would actually be more soluble in this mucus. Um, so if we had a cell or a drug that maybe was in this basic form for whatever reason, it could get into this mucus pretty easily, but then it might clear, right? The mucus might push it out. Let's say this is like the nostril right here. It might leave our nose. But if we also had an excipient, which could open these pores more, it could actually go paracellular into our body, as opposed to just being transcellular. Interestingly, um, for let's say it's in the just HA form, right? This lipophilic form, it wouldn't have a very easy time getting through this mucus, right? Because the mucus is very water-based, it's water-soluble, and this is lipophilic, right? So it wouldn't want to mix. So what you'd almost need is you need an excipient to kind of break up this mucus, like be a mucolytic, and then it'd be able to be transcellular. So it's almost like for nasal delivery drugs, you always need two things essentially, or in most cases you will. If it is an uncharged drug, you'll need like a mucolytic agent to go the paracellular route. If it's a charged drug, you'll need something to open tight junctions, so it can go the paracellular route or the transcellular route. I'm sorry, did I say that backwards? Lipophilic paracellular, hydrophilic transcellular. Sorry if I mixed those up. Um, Also, particle size to like stay in the nose and be a good nasal drug. Um, it needs to be smaller, right? So if we have a certain amount of drug that is kind of below a certain size, most of it will end up in the nose versus in the lungs. So to be nasally retained, we'll want it to be greater than 10 micrometers, but we'll also want it to be small enough that it can like go through these tight junctions if it's charged. It's the opposite for pulmonary drugs, and we'll get into that in a bit, but it will stay in the nose, right? Just because the nose is more accessible and the lungs can get really small really quickly, especially when you get to those alveolar regions. So that's why. You generally want these to be a little big. I think generally you'd want them to still be below about 30 though. So you can see here for this atomization device, the spray is 30. It has good coverage, and it can deliver things like naloxone. Here are the excipients. Again, I'm not going to talk too much about them because they are on the Quizlet, but I did throw them on there just because. This Optimus device might be a device we're asked on. This essentially makes it so the way it works is less drug will enter the stomach from the nose, because it has a way that it like closes up the trachea and the esophagus when you use it. I forget how. There's like a video on it in class. Just know that about the Optimus, that it's a really good nasal device. Just because you get more in the nasal region than in the lungs or in the stomach. Um, and then as far as like pharmacokinetics go for these nasal devices, um, you essentially don't want them to be cleared too fast, right? That's why we have the excipients to help like with blood flow or to help mucus flow be reduced or increased depending on what we need. And then there's a few ways these can be cleared besides just enzymes. It can just be cleared out by the mucus, right? It can expel, kind of like we talked about right here, where it went out the nostril. Um, and then there's also enzymes that can destroy it in the nose as well. Okay, so now let's talk about ocular delivery. Um, so nasal delivery, something that I believe I mentioned, but in case I did it, it you want it to be systemic generally, right? I mean, I guess you could have it be local if it was a problem only to the nose. I guess an example of that would be like oxymetazoline, right? An alpha 1 agonist to help with decongestion. That would be an example of a local, but you can also do systemic things like naloxone. The eye, you never ever want it to be systemic. So I would definitely remember that. That will probably be a question in some form when it comes to ocular delivery. The eye is also really complex in that it has a lot of barriers that kind of prevent drugs. It has two types. It has static barriers. These are more like anatomical based. So you have just the cornea, right? The fact that it exists is a barrier. Then also the blood aqueous barrier. Like there's obviously an exchange rate between the aqueous humor of the eye and the blood that's constantly flowing. Aqueous humor flows out, which is generally what's kind of preventing us from getting a drug deeper into the eye. For dynamic barriers, we have blood flow in the conjunctiva. It's kind of one of those outer layers of the eye. We have flow of lymph, right? Lymph is really important. It's connected to our blood system, but it will generally flow outward. And then probably the biggest one for dynamic is that tear drainage, right? When we have tears, they will kind of go out of the lacrimal gland and they'll come out of the eye. Now, here are the ways that drugs can enter the blood and be systemic, which is what we don't want. There's three primary pathways. The biggest one is probably this nasolacrimal pathway right here. This is the nasolacrimal duct. It's like if we almost flow down this, all of a sudden we're in the blood, we're in the body. Not good. The other ways are the conjunctiva. You might remember this was also a dynamic barrier. So it's almost like you can see they're overcoming these dynamic barriers that can essentially go deeper into the eye and work their way into the blood. Um, Same with the cornea. This was a physical barrier, but the deeper you go, eventually you can be in the blood. And the way that this would happen, right, is the drugs will diffuse more if there's a higher concentration. So these are generally lower concentration drugs to avoid getting into the systemic circulation. Um, Eye products are really strictly regulated in what they're able to do. Um, so one thing is they must be sterile. Um, there's two types. There's terminal sterilization type or versus aseptic processing. Terminal sterilization would be like you heat the drug up in a really hot oven to kill any bacteria before you, like after you've made it. Terminal meaning at the end. Aseptic processing is a lot harder to do, but it essentially means your entire process is sterile. So you wouldn't need that last step, but it makes the rest of your experiment a lot more, or not experiment, I'm sorry, your manufacturing a lot more difficult in order to do. Preservation's really important. It's only required for multi-dose units. All products might have it regardless generally, but essentially the idea is, right, if you open something, you don't want it to like rot and decay between the next time you use it. So that's why it's in multi-dose systems. Um, Basic things, isotonic, right? It'd burn the eye otherwise, buffering. You don't want to put like acid or strong base in your eye, it would blind you. Um, you want the particles to be really small. You don't want the clumps of stuff in your eye, especially because it'll clear easier, but it'll also be uncomfortable. Less than 10 micrometers is pretty standard. Um, and then viscosity can vary. Higher viscosity will increase contact time. So something like methyl cellulose could do that. It's not required, but it could help your dose form just depending on what it is. Um, as far as disease states you can treat with topical eye products or ophthalmic products is glaucoma is the biggest one. We did this all the way back in PPI. I've got a video on that. I don't really think you need it for this course, but if you want to get my watch time up, you can go watch that. Um, what are we trying to do with glaucoma drugs? So glaucoma at a basic level is just an overproduction of aqueous humor. And right, this can eventually lead to a pressure buildup and kill the optic nerve, leading to blindness. That's all glaucoma is. So how do we decrease aqueous humor secretion? Two ways that are really easy. We have anticholinergic drugs, right? Because if you remember, I have PPI videos on those as well. Anticholinergics will decrease secretions. Another way to do that is adrenergic agonist. So things like epinephrine. So And some antihistamines will just lower this production rate, and it'll make it so the eye doesn't produce as much humor. There's some carboxyesterases in the eyes that will kind of break down drugs like dipivarin into epinephrine just over time with how it's metabolized. So you can have prodrugs for more of a sustained release. Um, a couple of different ways. Just know that for glaucoma, you're just decreasing secretions. Um, so different kinds of ophthalmic drugs. You have solutions. These are kind of nice in the fact that they're very liquidy, right? They're in your eye. They're pretty easy. But the problem is, you really can't have too much of these, just to not be overwhelmed by like the max kind of amount you can put in the eye, those 30 microliters. And then your contact time's really short because while your eye is producing tears, you're kind of like washing it away. So if this is something that's going to act really rapidly, honestly like an adrenergic agonist, something that's going to work like right away, right when it makes contact, these are kind of fine. Uh, but like you have to also hope your drug will bind to its target for a longer time once it is bound. So it's kind of like not super practical. You need to administer it frequently, and then that might decrease adherence. Suspensions are a little better. They can be irritating, and they might lack some physical stability, right? This is when you need like a preservative for your multi-dose products, because the suspension, if you remember, is kind of like something insoluble, kind of like floating around in liquid, and that over time can decay pretty easily because it's not like a permanent thing. And steroids would be an example of like this. And then the one that has the best contact time that's not really going to get washed away are ointments. The only problem is they impair vision a lot, right? It makes everything really blurry because you've essentially got like a paste on your eye. Um, and like petrolatum would be a good base to like make something like that, but it's generally not good. Something like polyethylene glycol is a little bit better, just because it's clear, unlike petrolatum. But yeah, none of these are ideal, right? It's like you don't usually only, in fact, you'd always only use an ophthalmic product if it was something to do with the eye. So even if like let's say we wanted to give something systemic, let's say we break our golden rule of ophthalmics and we try and give it, the eye just is bad for it. You'd have to do so much drug and it'd impair your vision and it'd be so many problems. Basically just use ophthalmic products to treat eye conditions. That's why we do it. Um, what are some other ways we can deliver stuff to the eye? This will be really similar to if you remember kind of transdermal delivery. A lot of these are the same. So we have the microneedles, right? You can put like cyclosporin on them, inject into the eye. Again, these needles aren't painful because they're so small. It's like getting a mosquito bite, so you wouldn't really feel it. But I don't know. It's still weird. Something more specific to the eye are what are called punctum plugs. Uh, these are essentially like biodegradable plugs with drug in them. They're inserted surgically into your tear ducts. So this kind of eliminates two problems at once. You have a sustained release of drug into your eye, but you also don't let your tears clear it because you're blocking that lacrimal gland, or that lacrimal tube, whatever you want to call it. So it doesn't get washed away and it's sustained release. So these are actually pretty good. It's just obviously you need them placed by a medical professional, not always the most practical. Um, Something else, are there different types of like inserts? You have like this implant that's it goes under the conjunctiva. Uh, these are biodegradable. They're really nice. You can see they're not even like a centimeter long. They'll deliver drug for about a week and then degrade. Kind of annoying because again, like most of the time you would need these surgically implanted. So it's not super convenient to go to a doctor's every week, at least not for most patients. You've got something like Occusert, which are kind of like layers of different things. There's like a membrane to control release. There's the actual drug reservoir, and this would also be used for like glaucoma. It's pilocarpine, which is an anticholinergic that can be used for glaucoma. Um, yeah, so that's the time you might see that. It's fine. It stays around for a little bit. Something a little more invasive, but a little more effective long term are like these intravitreal implants. Essentially, there's like some drugs. These will be inserted into like kind of the core of your eye, but they'll last for like 3 years. So it's like surgically, yes, it's a little more complex than even something like microneedles or some of the other methods I've talked about, like the punctum plugs, but it does last a lot longer. There's also iontophoresis. If you remember, this is like where you apply a charge to a membrane. You electrocute your eye basically to get a drug across. You won't feel it, but like again, just weird going hand in hand with the microneedle. There's also intravitreal injections, right? Injecting like fluid or devices into the eye, into like the core of the eye to have that release. And there's also medicated contact lenses, which are nice because they kind of provide that sustained release and they're not really invasive. So those are pretty nice. Next up, let's talk about pulmonary. So when understanding pulmonary, I think the most important thing to understand is kind of the anatomy. And it's the fact that eventually when we breathe in, right, things begin in the trachea, then they go into the bronchi, things get smaller, and then they get really small by the time we're in the alveoli and alveolar sacs. Some of the cells are important to realize as well, right? Higher up, we'll have a lot of ciliated cells and goblet cells. Right, if you remember goblet cells, they make mucus. And ciliated cells will kind of push that mucus up. The reason this is important is because if it's like a really big drug, it can kind of get caught in what's called impaction. Um, well, impaction would just be it's too big to get to like the next part of the lungs. Sedimentation would be like it gets caught in the mucus, right? It gets caught in these bronchi, and the mucus is expelled outward, right? Because the whole point of our lungs is to kind of not have solids in there, or liquids for that matter, just to have gas exchange. So it'll kind of push those out. So that's what bronchi focus on. Bronchioles, still some ciliated cells, but we're not really producing that mucus anymore. We're basically just trying to get things out that aren't there. And by the time we get to alveoli, the gas exchange cells, we just have pneumocytes, which are solely for gas exchange. Um, when it comes to sizes of what drugs will kind of pass through, to get to our alveolar region and to be a good pulmonary drug, you have to be less than 3 micrometers. These tubes are really, really small. Now there is absorption in like, let's say the tracheobronchial region. And you can have up to 10 micrometers there, which if you remember, this was also kind of our cutoff for nasal delivery. So it's like once your drug gets through nasal, this would probably be where it gets cut off if it's less than that 10 micrometer, but greater than 3. But there's not as much surface area down there. The really nice thing about the alveoli, which makes them so good for gas exchange, there's so many with such a high surface area, but it allows us to absorb drug really quickly as well as absorb air really quickly. Um, yeah, and then if you're greater than 10, you're just in like the trachea or the nasopharynx. You're really not a good pulmonary drug at all. Um, I mentioned this before, impactions when it's too big to get through the tube. Sedimentation is when it gets stuck in the mucus and expelled. The last kind of thing is Brownian motion. This is more when a molecule is so small. Forces of like fluid and convection will kind of like push it around in different directions. It doesn't necessarily mean the drug will be like cleared or anything, but it makes absorption harder if it just goes in random directions. It doesn't go immediately down like gravity wants it to. It's a pretty minimal effect, but it is something to be aware of with really small molecules. Um, Ways drugs can escape are two ways really. Uh, they can escape by being cleared, being pushed back up by mucus. They never enter the system. That mucus might also be reswallowed and go into the stomach instead, where the drug is useless. And then also macrophages might kind of phagocytize these molecules. They'll like swallow them up, and then they're not any good to the body. Alternatively, though, we can have absorption, which is another method of escape. Kind of escapes from the lungs into the blood. A few ways this can happen, just regular passive diffusion, right? This is how most of our drugs work. There's endocytosis, right? Cells can engulf these, but not in the same way macrophages do, in a different way that'll actually get them to the blood. Or transcytosis, penocytosis, they can like follow water or small molecules. As far as like dose devices, there's a few different things that you might be familiar with, or you should be familiar with from asthma and therapeutics. We have nebulizers, right? Really big, really bulky devices. It's kind of nice because it occurs over a long amount of time. You don't need to breath activate it. Um, but you do need kind of that patient coordination, or I'm sorry, I'm looking at meter dose. This is out of order. Nebulizer. Okay, let's start over. So you don't require patient coordination. You're able to do high doses, but they're expensive, they're big, you gotta prep them. They're not practical to take with you. Um, we'll go back to meter dose inhaler now, because that's the third one or the second one I have here. Uh, they're non-breath activated, which is really good. Something about meter dose is they will have fluorocarbons, so carbons attached to fluorine atoms. Those are propellants, which will kind of like shoot it into the lungs. This can be really good. You do need coordination to like time that though. And these are also probably the least efficient form of drugs. Dry powder inhalers are actually better than meter dose inhalers, and I'll talk about that more in a second. But this might be a question in some capacity, so I would remember that. Finally, dry powder, they're breath activated. Which can be good or bad depending on patient coordination. Um, there's no propellant. Instead of propellant, DPIs will actually use like sugar molecules to kind of like carry the drugs into the lungs and get them where they need to go. Harder to do high doses, right? Because if you do too much powder, the patient's gonna like feel like they're choking. A lot of deposition, a lot of this sedimentation we talked about before. Um, and then you can't use with like a trach tube, which like duh. But yeah, when it comes to these, um, meter dose inhalers, because of those propellants, they're a little bit, it's harder to know where they're going to be kind of shot into your either esophagus or into your trachea. So generally, meter dose inhalers, more of the drug that you want in your lungs is going to end up in the stomach instead. Um, versus dry powder, the powder is just better at with the sugars and stuff getting into the lungs for one reason or another. Um, so it's got some higher efficacy in that regard. Although there is the drawback, some patients just can't do the dry powder because they feel like they're choking, which isn't something that doesn't help adherence, to say the least. So yeah. Finally, I want to finish up with biologics. There's a lot in this a slide deck, so I'll go through it at a very 1,000-foot overview. I don't think these questions are going to be the vast majority of the exam because there's not a whole lot of application. It's kind of just memorization, but I could be wrong. So I'll go through it really quickly. What can be biologics? You have things like blood components, antibodies are probably the biggest one, um, cell and gene therapy, uh, tissues, proteins, really anything that comes from an organism. Um, One of the things I do think will be asked is some insulin modifications. So for example, detamere has this fatty acid chain. I would remember generally, they mentioned in the recitation that they might ask a question on this. They said they wouldn't ask like what amino acid residue things were on, but what was added. So for example, you could say detamere has a fatty acid attached to it. And then I didn't want to include every one on those because I don't think it's super relevant. It's kind of just memorization and it's in the slides, but that might pop up. To be honest, I think it is going to be a Detamere question because I know that was a practice question. Something tells me they just reuse it, but I could be wrong in that. Um, and then what generally differentiates like biologics from just small molecule drugs? Biologics are really big. They're 150,000 Daltons versus like you remember Lipinski rules of 500. Um, there's a lot of aspects to productions, right? There's a lot you need to do. They're species selective because you might have like a reaction using a human antibody in a human. They're multifunctional, which is really good. They can do a lot of targets, things of that nature. Um, as far as toxicology go, it's usually on target toxicology, meaning we have like too much of a response in one area, whereas with small molecules, they might eventually like bind to something else. Um, they're in the body for a long time. That can be pretty good depending on how we dose them. So infrequent dosing, but again, that's usually like injectable dosing as well because you can't really swallow these. So give and take. Their PK is linear, which is pretty interesting, all things considered, because most of these are like first order, um, and that can be like annoying just to calculate and to dose adjust for. Um, and then there is immunogenicity, which is probably the biggest drawback of these, meaning the body might have like an allergic reaction or an immune reaction, where small molecule drugs do not. So overall, monoclonal antibodies are actually pretty good. It's just right now they're really expensive because they're newer, or at least being more heavily invested in now, even if they have been around for decades. Um, but yeah, I think these drugs have a lot of potential. Um, as far as the PK of biologics, slower distribution because they're so big means they have more time in like the central compartment, right? More times in organs like the heart, the liver, the kidneys, these really highly vascularized organs, just because they don't get out of the blood very easily. They're usually proteins, right? And that's actually pretty good for them. It usually means they're just degraded via like proteases, things in the blood that just break down proteins over time. So you don't really get like toxic metabolites with these. You might technically, but it's so rare. Um, yeah, metabolized, not metabolized by CYP. I basically just said that. You have metabolized. They can actually be recycled via a pathway called like FcRn. This is when they enter a cell. They might eventually be sent out of a cell if they're not metabolized and be reused. That helps half-life. Um, if they target something, it kind of affects where they're distributed and cleared, right? If it accumulates in a certain tissue more than others. Um, and then immunogenicity, if your body's getting rid of it, this can actually lead to faster clearance than expected in some people, which isn't great. In others, it could be slower if they have less of an immune response. Yeah. So let's talk about some ways to kind of like produce antibodies. One way is by kind of introducing an antigen into an animal model, having the B cells, which you remember create antibodies, and then you can essentially farm antibodies by doing this, by removing it from this animal's blood, and then we can refine that into a drug. Another way is we can create what's called hybridomas. We inject special cells, which will essentially turn into something called a hybridoma, which are like specialized cells. And then from here, we can kind of further this process, select for these cells, and then farm more different types of antibodies. And since these are like combined cells, we can do more than one at once. And both of these are in vivo. There probably eventually will be in vitro ways we can create antibodies from cell plates, but it's just not quite at the point where it's viable yet. So we're still kind of using animals to farm these in the meantime. Um, the biggest way these will kind of like move are convective transport. These will kind of just flow around the blood. We kind of talked about that. They stay in the blood a really long time. They just kind of quote unquote go with the flow, I guess. Um, and they're eliminated via catabolism, right? That's breaking down big molecules with those proteases, and we just turn them back into amino acids. The actual structure of an antibody is the only part that kind of does anything are these antigen binding sites. We have this light change, which are these pink sections, and we have these heavy chains, or these FC regions. You might also see them written as, which are kind of just the backbone. And it's really these lighter areas that actually do anything. These are the constant regions. That's what the FC and constant stands for. There's some drugs that, well, they generally do look like this Y formation. Some drugs may just be like these fragments if it like needs to get into a cell or something. Or there might be like modified antibodies that look differently. So that's kind of a benefit of these. The most common like basis for our antibodies, they look like IgG. I believe this is an IgG antibody. I forget exactly which ones look like which, but I'm pretty sure it is, but feel free to correct me if I'm wrong on that. I don't think that'll be a question. Um, the average half-life of an IgG antibody is 20 days, right? So injections every like maybe a couple of months, right? Because like even one half-life, that still means half the drugs in your body. That's pretty good. Like if you did our rule of 95%, that would be like what? I think every 3 or 4 months you need an injection. So overall, like that's really good versus taking a pill every day. I feel like even if you don't like needles, I feel like that's reasonable. Um, there's different naming schemes for antibodies. OMAB with no FC region, so none of this region. That would look like one of these things that I circled. That would be an OMAB drug. Um, ZMAB is a chimeric antibody. Um, that just means there's some human parts and some other animal mouse. That's or animal part. It's usually a mouse. Humanized would be Zumab. This would be an antibody from another animal that we've inserted human parts to, and we've made mostly human, but there's still some animal components. And then Umab is actually a human antibody itself. And these ones have the highest chance of like immunogenicity. These are probably the lowest. And then I actually don't know about these just because they don't have that full region. It probably varies, to be honest, because I'm sure even these can be combined with different animals in some regard. But this is like the highest chance of an immune reaction, and this is like the lowest. So that might be asked in some capacity. And then the final kind of lectures are on like personalized and precision medicine. Precision medicine is essentially something that will focus on the disease to make sure the disease is understood, diagnosed, and then treated individually. Personalized medicine is more of like predicting or treating a specific patient. Yeah, it's like treating a patient's specific needs. That could be genetic based, that can be social determinants of health, that can be a lot of things. Predictive medicine is used to predict the probability an individual will get a disease, whereas preventative medicine is to kind of prophylactically prevent diseases when we can predict they're happening. And therapeutic medicine is used to treat diseases that a patient does have. And that's almost always through pharmacotherapy. Um, when it comes to individuals, the risk of developing a disease, the idea is since we can predict it based on genetics, we can minimize it. And a disease can be treated based on, it says molecular information. It means like DNA or like genetic code, right? Based on your genetics, we know this is going to happen, and based on genetics, we know what medicines will work the best, or at least that's the goal of personalized medicine, which is a pretty tough goal. We're getting better at it, right? Because I'm sure you remember from MMP and other classes, some populations may express CYP enzymes differently, which we can dose our drugs differently, and then different enzymes being elevated or lower might make you more susceptible for like a disease. So it's like we're pretty early stages with this still, but if we could do this, this could obviously help healthcare a lot. Um, so what are some challenges? They kind of fall into the ranges of like technical to social, and then there's also just the difficulty in general, right? So it's like, getting support is more of a social thing. It's like when you think about like COVID vaccines, right? But even though they were very readily available and we got through all these technologies, like I think 30% of the population still didn't want to take them. So it's like, well, we did have this thing and it was very technically challenging to make. We couldn't necessarily get the public support we needed for it. Um, versus like something like using computers to like predict medicines, right? The public doesn't care. The public's not going to be adverse to that. At least not yet, knock on wood. But that's very technical to use like AI and stuff to help us create medicine. Um, a few kind of things about just random drug examples we might get asked because these were both on the slides and they were also in the quiz is Tamiflu. If it's not hydrolyzed, it might form this toxic metabolite. Um, so that's bad, except clopidogrel, if it is hydrolyzed, it will form its more active metabolite, um, I believe. Or maybe it's the inactive one. Let me get back on that. I'll like re-report the slide at the very end. So we'll address that. I need to double check that. But and something I do know about clopidogrel is how it interacts with aspirin. Aspirin's really interesting, right? Because it is a competitor inhibition of CYP enzymes. And if you remember, clopidogrel is a prodrug. So if we're inhibiting an enzyme that would metabolize the prodrug into the active, aspirin will lower clopidogrel activity. And the most common thing it inhibits, it's CYP2C19. Or no. Yes, CYP2C19. But aspirin also induces other CYP enzymes, like CYP1A2, I believe, which clopidogrel is also metabolized by to a lesser extent, but this would increase its activity. So aspirin is weird, and that's why aspirin you really need to dose it carefully with clopidogrel, just to kind of, or like at least monitor, because I guess the dosing is always the same for like prior MI and whatnot, but yeah, just something to be aware of. So that's pretty much all of that when it comes to specific enzymes. Now something else, in order to improve how we are moving medicine forward, um, we need to like keep in mind like diverse populations, right? To understand these genetic differences and then account for them, we need large studies, right? More data, more AI, use pharmacogenomics more, utilize health records more, be sustainable to the environment, and then retain the trust of the public. These are all kind of common sense things, but if we had all 7 of these things, we would probably be able to push these medicines even further. Um, Something else is toxicity can essentially most of the time be determined genetically, right? This is especially true when it comes to um, things that CYP enzymes are metabolized by. Um, yeah, so it's like the whole idea is that we can determine what drug is going to be toxic to anybody when we get genetically advanced enough or when we understand genetics enough. Um, Difference between dosage and dosing. A dose is a specified amount of medication given at one time. Dosage is the complete term, amount, frequency, all that. And then some studies for kind of monitoring drug efficacy are there's 2 kinds. There's PK, PD studies, so pharmacokinetic, pharmacodynamics. There's also PBPK. I forget off the top of my head what PB is, but essentially, since there's no pharmacodynamics in this one, the big takeaway is that you would not have an efficacy test because pharmacodynamics essentially is efficacy of a drug, where PBPK would just look at kinetics and then something else. And then generally something else is PK/PD studies are more complex than PBPK studies. Okay, I went back and I looked again at the Tamiflu and clopidogrel. So Tamiflu is actually activated by hydrolation. It just also makes it toxic as well. So that's something to keep in mind, that the active form is actually a little more toxic than the inactive form, but again, like it treats the disease, so we kind of take that risk. When clopidogrel is hydrolyzed, it becomes inactive, which is kind of just right here. I don't know how I missed that on the slide before, but whatever. It's there now. So with that, thank you so much for watching and good luck on the exam.